Hello, everyone, and welcome to today's broadcast, Accelerating Tuberculosis Elimination with Genomics, presented by Dr. Jennifer Gardy, an Associate Professor and Canada Research Chair in Public Health Genomics at the University of British Columbia and a Senior Scientist at the BC Centre for Disease Control. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appear on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing the presentation, just click on the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of your presentation window and follow the process to obtain your credits. Please join me now in welcoming our presenter, Dr. Gardy. I will now turn the presentation over to her. Welcome, Dr. Gardy.
Thank you, Dr. Valdez, and it's a pleasure to be here speaking to you all today about some of the work that we're doing up in British Columbia, Canada, on using whole genome sequencing as a tool to accelerate our progress towards tuberculosis elimination. Now, for those of you who might not be familiar with TB, I thought I'd start with a bit of a recap. TB is something that people often say, well, why are you still working on that? I thought that was a disease of the past, but in fact, the opposite is true. TB is actually the #1 infectious disease killer worldwide. It surpassed HIV a long time ago, and in terms of a single infectious disease, it is the #1 killer, despite the fact that we have medication to treat TB. There's a number of different antibiotics that are effective at treating the organism. When we talk about tuberculosis infection and disease, we're talking about two different things. Latent TB infection, which we often abbreviate to LTBI, is a type of TB infection where the TB germ is in your lung. It's been walled up in a little kind of a little ball called a granuloma that your immune system is created and kind of keeps under control. And latent TB infection isn't transmissible. It doesn't cause any symptoms. You're not actually sick with any TB disease. In fact, about a quarter of the world's population is actually estimated to have latent TB infection. Only about 1 in 10 of those individuals, though, will ever go on to develop active tuberculosis disease. Active TB disease is when the TB bacterium escapes from that little ball, the granuloma, starts replicating in your lung and actually causing symptoms. So transmission of TB only occurs from active TB disease cases, people that are symptomatic and infectious, and transmission is through the airborne route. Along with measles, TB is one of the truly airborne pathogens where you could cough some TB bacilli into a room, leave that room, and then somebody that comes in a short time later might inhale those and receive the TB bacterium. There is a spectrum of infectiousness in terms of how likely people are to transmit TB to another. TB can really cause disease anywhere in the body. We do primarily think of it as a lung disease, but it can cause infection pretty much anywhere. So people that have what we call extrapulmonary TB, where it's not in their lungs, they tend not to be infectious. They don't transmit onwards. And even amongst people that do have the pulmonary form of TB, there still is quite a range of how infectious they are, largely depending on how many TB bacilli they're coughing out. And if you pay any attention to the TB world at all, you probably are aware of the fact that antimicrobial resistance is an emerging issue that we're dealing with. We often talk about drug-resistant TB or multi-drug-resistant TB, where you've got resistance to multiple TB antibiotics, as well as increasing rates of what we call XDR, or extremely drug-resistant TB, where you're resistant to virtually everything that we could treat your TB with. So this is an emerging issue in TB. MDR TB is arising de novo in people that have difficulty completing the course of antibiotics that they've been given for whatever reason. And we're also seeing transmission of drug-resistant TB. And an interesting thing about antibiotic resistance in tuberculosis that's different from a lot of other microbes is that resistance is driven by point mutations. It's single nucleotide changes in the TB genome that lead to those resistance phenotypes. TB is unevenly distributed around the globe in terms of TB incidence rates or the burden of disease. You can see the bluer the country on this map, the higher the TB rates. So in places like North America, our TB rates tend to be quite low, generally less than 10 cases per 100,000, typically closer to something on the order like 3 to 5 cases per 100,000 population. But in places like Southeast Asia, Sub-Saharan Africa, rates are quite high, in the hundreds and hundreds of cases per 100,000. And because of the way that migration works and the way the world is these days, we often say that TB anywhere is TB everywhere. So people that are migrating from higher incidence countries can often bring that latent TB infection. Remember, a quarter of the world's population might have LTBI. They can bring that infection with them to other parts of the world where eventually it might go on to wake up and become active disease. So in settings where there's fairly good laboratory infrastructure, you've got TB labs, reference laboratories that are responsible for all sorts of aspects of the laboratory investigation of TB, from diagnosing a TB infection. There's also a lot of other mycobacterial species out there beyond Mycobacterium tuberculosis that causes TB. So a mycobacteriology lab is often trying to differentiate, does this person have Mycobacterium tuberculosis, or do they have one of the NTMs, the non-tuberculous mycobacteria that don't cause TB disease? Laboratories are also responsible for phenotyping. So they're doing antibiotic sensitivity testing of isolates that come in, and they do genotyping as well, DNA fingerprinting to determine whether a newly diagnosed isolate is perhaps part of an outbreak and related to cases that we've seen before. So currently, some of the molecular tools that are used, there's some very simple tools. A lot of diagnosis just happens via culture, but we do some molecular work as well using PCR for diagnosis. And then the genotyping technique that we use in order to DNA fingerprint TB isolates, the classic genotyping technique is something called Miru VNTR. I often just refer to it as Miru for short, and that stands for Mycobacterial Interspersed Repetitive Unit Variable Number Tandem Repeat. So it's a series of 24 repetitive locations around the Mycobacterium tuberculosis genome that we amplify up with PCR, run on a gel, and we can estimate the repeat size, and that gives us a very simple way of representing a DNA fingerprint. So this is what your classic TB lab might look like, and that's what I have access to at the British Columbia Centre for Disease Control. So this is where I work. This is it lovingly rendered in watercolor painting, and it's a really interesting place to be a scientist because we are the provincial health agency. We do handle everything related to communicable disease, laboratory work, surveillance, epidemiology for the entire province of BC. So we've got very extensive sample data. We've got all of the TB culture from all of the cases of TB diagnosed in BC, but we also have a lot of rich information about the patients and the populations and the communities that those isolates are coming from. So when we want to study TB from a combined molecular and epidemiological perspective, we're really tremendously lucky in that we've got access to this wonderful array of data. So in British Columbia, our tuberculosis rate is generally around sort of 5 to 6 cases per 100,000 population. And that's a rate that is slightly higher than the Canadian average. And that's largely due to the fact that British Columbia is a province that receives a lot of immigrants from other countries, particularly countries around the Pacific Rim. And we know that in places like China, like Vietnam, like the Philippines, there can be very high TB rates. So we do get a lot of people that were born outside of Canada in a higher incidence setting moving to British Columbia at some point in their time here, their TB progresses from latent to active disease, and that, I think, accounts for our slightly higher case rates than some of the other provinces out there. In terms of what that translates to in actual numbers, we typically see around 250 cases of TB in British Columbia each year, and the vast majority of those are in persons born outside of Canada, approximately 80%. And one of the nice things about TB in our backyard is that drug resistance isn't really a big issue for us. 90% of the TB cases that we diagnose, those isolates are fully sensitive to all the frontline antibiotics. There's only a very small handful of cases that are resistant to a single antibiotic, and an even smaller handful of cases that are resistant to multiple antibiotics. So we seem to be in a good position. That's not a lot of cases. Everything seems fairly manageable. However, we're not on track to meet the TB elimination goals that were recently set by the World Health Organization. So TB elimination is defined as having only one case per million population, and pre-elimination is defined as 10 cases per million population. So right now, we would love to be able to meet those goals in 20 years from now, in 30 years from now. But if we look at the current trajectory of declining TB case rates, this time I've put those in per million population instead of per 100,000 population, we can see if we do a little extrapolation, we're not on target to meet those goals. So obviously we need to be doing something different. A few years ago, a graduate student of mine and I wrote this article for a European Respiratory Journal. They had talked about, in an earlier ERJ article, the framework, the WHO framework for TB elimination in low-incidence settings, places like British Columbia, places like North America, had been laid out in this journal article. And we read it and realized that something they didn't talk about in the article, but that we felt was a particularly promising approach to really changing the way we approach TB elimination is genomics, and specifically how we can use genomics as a tool to both accelerate our time to diagnosing an individual with a TB infection and getting them initiated on the appropriate antibiotic therapy, as well as a tool for helping us to understand the epidemiology of TB transmission in a region and use that information to stop onward TB transmission. So this article kind of forms the basis for a lot of the work that we've been doing in British Columbia recently on incorporating genome sequencing into our routine tuberculosis prevention and care programs. So in BC, we've set our own elimination strategy. It's been ratified by the province, and it includes a couple of different things. As I told you, the bulk of TB in BC is in persons born outside of Canada. So in this population, what we really need to do is improve our screening for latent TB infection and get people that have been identified as having LTBI onto therapy as soon as possible so we can clear that infection and they're never at risk of progressing to active disease. So that's more of a programmatic issue. But where genome sequencing, I think, can help with this particular approach is when an individual does progress to active TB disease, can we rapidly diagnose them as having drug-resistant TB? Most of our drug-resistant TB and virtually all of our multi-drug-resistant TB does happen in persons born outside Canada. They acquired their drug-resistant infection overseas. So it really would be beneficial for us to be able to very quickly identify that these people have drug-resistant TB and get them on the right antibiotic therapy right away so that they become non-infectious and they're not transmitting onwards. And you'll hear more about that in a little bit. The other group that we're dealing with in British Columbia are people born inside of Canada, where TB is transmitting from person to person in the province. And when we look at what's happening there, really that transmission is happening in the most vulnerable of our Canadian-born population. Oftentimes these are people that are living on the streets or are transiently housed. So in this population, if we're to reach TB elimination, we really need to understand sort of where, why, and how TB is transmitting in these groups. And that's an area where genomics holds tremendous promise. So what we've done over the last, I guess we've been working in this area since about 2008, 2009. So over the last decade, as we've been thinking about how to incorporate whole genome sequencing into our practice, we've gotten to the point where we've now sequenced over 2,000 TB genomes that have been collected going back 13 years now. And we're looking at how can we use that information to improve our drug resistance prediction, and how can we use that information to track TB that's transmitting in BC and around Canada. So how did we end up with all these genomes? What's kind of the history of TB genomics in British Columbia? So the timeline looks a little bit like this. So as I said, we've been working in this area for about a decade. In 2008, we were funded to do a project called the 40 TB Genomes Project. And this was at the time when next-generation DNA sequencing technology was just starting to become available to the research community. And the notion of sequencing 40 complete genomes from a single bacterial species representing a single outbreak of infectious disease seemed a little crazy at the time, but it was a challenge we were willing to take on. So we went ahead and started that project. In 2009, we got the sequence data. We were only able to get sequenced for 36 of the 40 genomes that we had originally hoped to do, but that was enough for us to begin looking at how can we tell what happened in a TB outbreak from genome sequence alone. And you'll hear more about how we do that in a bit. But that led to a paper in the New England Journal of Medicine that was published in 2011, where we really sort of for the first time demonstrated that genome sequencing is a very useful tool for epidemiology, for reconstructing outbreaks. And that you could quite feasibly figure out who transmitted to whom. It was really the first paper of its kind in that bacterial genomic space. And at the same time that paper was being published, we also embarked on a second outbreak investigation. The one that we had started in 2008 was looking at an extremely large outbreak of TB in one particular region of BC. And in 2011, we started working on a second outbreak, which eventually came to be the biggest outbreak of TB in the province in recent years, and we looked at that with genomics as well as part of the real-time response to that outbreak. Based on our work looking at a couple of these TB outbreaks, we managed to convince our financial overlords to give us money to go and sequence all of the TB in BC. So we launched a study that we call the Molecular and Genomic Epidemiology of TB in BC. And so what we did with that was go back and retrospectively genotype every single case of TB we'd seen going back to 2005. So we genotyped it with miR-VNTR DNA fingerprinting. And what was cool about that is as we were analyzing those results in the early days of the project, they were so promising that in 2014, the TB lab decided just to implement routine miR-VNTR genotyping for all isolates that were coming in. So by the time we reached 2016, we were at the point where we had genotyped about 2,300 isolates, every culture-positive case of TB in British Columbia diagnosed between 2005 and 2014, so complete decades worth of cases, and we whole genome sequenced 1,400 of those. We were really interested in studying disease transmission and resistance, so we sequenced preferentially two types of isolates. We sequenced those isolates that shared a DNA fingerprint with at least one other isolate in the study cohort, and that would suggest that potentially these could be recently transmitted isolates. And we also sequenced all of those isolates that we had in the laboratory measured a drug resistance phenotype for. So where we're at these days is we've switched from sort of selective sequencing that we did in that retrospective study where we just sequenced the transmitted isolates and just sequenced the resistance isolates. Since 2015, we've been sequencing all of the isolates. If it is culture positive, it goes on the sequencer. And so we've completed a lot of the analysis for the 2005 to 2014 data, which is what I'll show throughout the rest of the presentation. And we've got a lot of really interesting data coming down the pipeline that we're currently analyzing from 2015, 16, 17, and now 2018. So the first story I'll tell that we were able to derive from this incredibly useful set of these thousands of TB genomes is the story around how can we accelerate people's diagnosis of TB, and in particular, how can we accelerate getting them on the right antibiotics therapy? So let's do a little imagination exercise here. Imagine that you are a doctor. Imagine that you're a doctor who also happens to be a cat, I guess. But imagine you're a doctor and you have a patient that you suspect of having TB. The timeline for that, you would take a sputum sample from your patient. You'd have them cough into a cup, and that sputum would arrive at our provincial reference laboratory. Ideally, the day it's received or the day after, it would be gram stained. It would go on to microscopy, and we would look to see whether we see any TB bacillus on that microscope slide. And we might get an answer that says no, or we might get an answer that says yes. So if we did get the answer that said yes, we would do a very quick PCR analysis with 2 primer sets, one that's designed to look for Mycobacterium tuberculosis and one that's designed to look for Mycobacterium avium complex. This is a non-tuberculous mycobacteria. But those 2 mycobacteria represent the vast majority of samples that come into the lab. So if you were lucky and there was bacillus visible under microscopy and there was a positive PCR result, you might get a preliminary diagnosis reported back to you within the space of maybe 1 to 4 days. But if you don't get a preliminary diagnosis, and even if you do get a preliminary diagnosis, we still do put that sample into culture. So negative results in all samples really go into culture. And then we wait, and then we wait, and we wait some more. And at some point, typically the fastest this might happen would be, let's say, about maybe 7 to 10 days after a specimen is inoculated into culture, we might get a result saying that something is culture positive. But Mycobacterium tuberculosis is a very, very slow grower, and it might be as long as 4, 5, maybe even 6 weeks before we get a culture positive diagnosis back and we can say, yes, your patient has TB. So at that point, we've given you a diagnosis. You might have been waiting many weeks for that. And then we re-inoculate that sample into culture, this time in the presence of antibiotics, and we wait, and we wait, and we wait, and eventually we get the results of our drug sensitivity test. So we've tested frontline antibiotics, and we should be able to then say, what do you have in terms of a sensitive or a resistant organism? So at that point, if there's any drug resistance observed, we then inoculate it into culture again, and we wait, and hopefully we get the results for any second-line antibiotics. So if you're resistant to one of the first-line antibiotics, we want to test your sample to make sure it's not resistant to any second-line ones, and you have MDR-TB. So as you can imagine, it's a pretty long process getting somebody to the point where we've got their drug sensitivity results and their second-line drug sensitivity results. So that's clearly a long and drawn-out process. If you want to get an answer of what does this patient have and what antibiotics I should be treating them with, you're often waiting many, many weeks, sometimes even months as a clinician to get your answer. So together with an international consortium led by Oxford University, a couple years ago we were interested in asking the question, could a single whole genome sequencing analysis replace all of those other analyses and, more importantly, get you a result, an accurate result, faster? And the long story made short, the answer is yes. We published this in Lancet Respiratory Medicine in 2016. And what you've seen is based on these pilot data, Public Health England, the National Public Health Agency in the UK, has now switched to routine whole genome sequencing for tuberculosis. And you're also seeing that happening in state and national labs here in North America, including the New York State Department of Public Health. So the premise behind whole genome sequencing as a tool for TB diagnostics is fairly simple. What we do is wait for a culture positive isolate to be positive for 1 day. So we still need that culture step. We inoculate a specimen into culture and wait for it to ding positive. Once it's been positive for a day, there should be enough sequenceable bacterial material in there in order to actually get genomic data out. So what we do is batch a few, typically 12 to 15 bacterial isolates, TB isolates that we've got off culture, and we sequence them, often on the Illumina MiSeq. Once the sequencing is done, the raw sequence data is uploaded to a central server where it's analyzed, and a report is returned to the clinician, to the laboratory, that shows the results of that particular analysis. So what we've seen with that, one of the most interesting things is looking at resistance prediction. So as I mentioned off the top, when we look at the TB genome, resistance phenotypes are encoded by single nucleotide changes, point mutations. So the idea of resistance prediction in silico basically says if we have a catalog of known resistance mutations, can we look for those in the genome that we've sequenced? And from that, if the mutation is present, can we reliably predict a resistance phenotype? So together with that Oxford consortium and a group around the world, we have a paper coming out very soon looking at 10,000 TB genomes, 1,400 of which come from British Columbia, and saying, can you reliably predict resistance to common TB drugs like isoniazid, like rifampin? And it turns out that you can, with actually surprisingly good sensitivity and specificity. So this is really promising. So as I said at the top, typically we might be waiting weeks for drug sensitivity results to come in. On average in BC, we're waiting just about 6 weeks in total to get our DST results. But with whole genome sequencing, we can get those within a day or two of a sample showing up as culture positive. So we can couple resistance prediction and getting people on the right therapy to diagnosis. This is huge for accelerating the time to getting people on the right therapy. One other thing that we've been working on as a result of this collaboration with Oxford and with Public Health England is how do we avoid, when we present the results of this genome sequencing-based diagnosis and phenotyping, how do we avoid overloading people who might not have seen microbial genomic data before? How do we make the results clear to them and not overload them with too much complex genomic information? Because there's a lot of computational biology and a lot of genomics that goes into putting together a resistance report, putting together a diagnostic report. How do we make that information clear to people? And so in that pilot project, there was a very simple report that was designed. This is the report that we were using back in 2014 when we were engaged in this pilot project. But as the years went on and as the bioinformatics capability of analyzing TB genomes and deducing interesting things from them gradually increased, we found that the bioinformaticians who were generating these reports started to put more and more information on, and that information wasn't necessarily being communicated in the clearest manner. So the current version of the report that comes out automatically at the end of this sequencing and analysis pipeline looks something like this. And to somebody like me who's been trained in computational biology and microbial genomics, I can make sense of most of this fairly easily. But you can imagine a clinician, a TB doc, who's spent their entire life treating patients and has never seen genome sequencing before. This could be rather confusing to them. So some of the work that we've been doing really aimed at making TB genomic data more actionable, more useful to a broader audience, has been around communicating these results. So this is some work that was done by a graduate student and a medical resident that were working with me, Anna Krisan and Jeff McKee. And they did something borrowed from the field of information visualization within computer science, and it's the notion of a design study. If you want to create something that's going to communicate information, in this case genomic laboratory test results, to end users, The way to do that effectively is to figure out what those end users need. What are they doing with the data, and how can you visually present the data in a way that's going to make their life easier? So we did this design study process. I'm not going to go into this in great detail. There's an open access paper that you can read all about this. But we did find some interesting things. We found that when you're presenting laboratory test results, whether they're genomic or not, to people, there's a lot of constraints that are applied on that process. Clinicians don't often have a lot of time to review the report. They're often dealing with multiple different documents that are coming in at different times. So they'll get a diagnosis one day, and then they have to wait, weeks to get the drug sensitivity results. And the report formats that are used can be quite different. Some public health agencies are able to report results through electronic health records. Others still rely on faxing information back and forth. And depending who you are and your role in TB prevention and care, you might want to do very different things with the results of a TB genomic analysis. If you're a clinician, you're probably just interested in what does this patient have and how am I going to treat them. But if you're somebody working in public health, you're probably more interested in, is this isolate that I've just diagnosed part of a larger outbreak? Do I have something troubling on my hands? So we got together and surveyed a bunch of users. We did a little process that's referred to in information visualization as a design sprint, where you get a group of people around a table designing some different visual report prototypes. So we sketched those up manually. Then we turned those into electronic versions. We developed 4 different prototype reports. And then we did a type of experiment where we asked our end users to evaluate individual components of the report. We were looking at small little visual elements and giving them a choice between 2 things, saying, hey, when we present results in this way versus this way, which one do you like better? So, for example, one of the questions we asked is when we're reporting out the wording that we use to describe TB antibiotics, there's a couple different ways we could do it. We could use the drug's full name. Every drug has a 3-letter code. In TB, they also have a 1-letter abbreviation. So we asked people, both clinicians and non-clinicians working in TB prevention, which option do you prefer? And so we did this for all of the different design elements in the report, whether it was, drug names or the type of table that we use and how resistant isolates are highlighted within the table. Is there highlighting? Are they bolded? Are they shaded? Is there a little icon? We went through and did this for every type of visual element that was present on the report. Here's another example where we looked at how do we organize susceptibility and resistance results. And we ended up assessing a whole bunch of different factors, and based on people's preferences for particular design elements, we were able to arrive at a very simple final report that meets the needs of our users, that takes complicated genomic information, hey, we use genome sequencing to diagnose this as a TB isolate and to predict its resistance phenotype and to assign it to a cluster or potential outbreak and communicate it in a way that makes sense. And if you're interested in following up that report anymore, the URL to that report is available down at the bottom. So that kind of summarizes some of the work that we've been doing on the resistance prediction side of things, and I wanted to spend the last bit of time talking about that other piece. So part of accelerating progress towards TB elimination is diagnosing TB quickly, getting people on the right therapy quickly, but part of it is also understanding outbreaks much better than we do now. And this is Dustin Hoffman's 1995 film Outbreak. It's a terrible movie, but it's one that I saw when I was a teenager and said, that's what I want to do for the rest of my life. So let's look at how we investigate TB outbreaks. So right now, our tuberculosis lab is responsible for diagnosing cases of TB within British Columbia. So over the course of a month, you might get a whole bunch of cases diagnosed, and I've represented those by colored dots. Then with DNA fingerprinting, that mirror VNTR genotyping technique that I mentioned earlier, we fingerprint those isolates to try to figure out, are any of them related to each other? Are they genetically similar enough that we might expect recent transmission, person-to-person transmission? So here we said, oh, look, there's a bunch of cases that have the same fingerprint. All these blue cases might potentially be related. But in order to draw the connections between those cases, we need to do a contact investigation. We need to do field epidemiology where we go out and we talk to these patients and we try and understand who they have social contacts with, where they live, where they work, who do they see in those places, et cetera, et cetera. So this has worked decently enough for the past decade or so, but there are some challenges. Fingerprinting, DNA fingerprinting, only tells you that there's a cluster of potential cases, and it often overestimates clustering, as you'll see in a little bit. So it just tells you that cases are related, potentially related to each other. It doesn't give you any sort of directionality. If you want that directionality, you have to go in and do your contact investigation, do that field epidemiological work, and that can be challenging sometimes for a lot of reasons, not the least of which is that public health is fairly resource limited. We don't often have people that we can send out to investigate every single outbreak all of the time. And depending on the fingerprinting method that you use, you might end up with slightly different results. So most laboratories now are using 24 locus mirror VNTR analysis, but before that we used to use 12 loci, and I can tell you that when we switched from 20 to 24 loci, the number of outbreaks that we thought we had on our hand decreased because we had a higher resolution technique. So as I said earlier when I was recapping what we were doing with tuberculosis in British Columbia, we did that big retrospective analysis looking at all the culture-positive TB cases going back to 2005. And if we look at that complete decade of TB and their mirror VNTR genotypes, their DNA fingerprints, this is the diagram that we get. Each of these circles represents a different unique genotype, and the bigger the circle, the more people we observed that genotype in. They're colored according to birthplace. Genomically, we know that TB kind of falls into a couple different lineages. We describe these as things like the Euro-American lineage or the East Asian lineage. You can see that most of the TB in BC is Euro-American TB, and certainly most of the large clusters, those big nodes, occur with Euro-American lineages. The genotypes in the other lineages tend to be singletons. We only see them once. So this type of information can be very helpful to tell us how much potential transmission is there. And if you go just on genotyping data alone, you would estimate that in British Columbia, 42% of our isolates are clustered. So potentially 42% of cases are the result of transmission happening in BC. But there's a problem with that, and it's that resolution problem that I mentioned a couple slides ago. Mirror VNTR is only looking, best case scenario, at 24 variable number tandem repeat locations scattered around the MTB genome. It's not looking at all of the available information. So when you're doing genotyping, I like to explain it as it's like you're looking at a picture, but you've taken 99% of the pixels of the information away. You get a rough idea for what the image might be, but it's only when you look at 100% of the pixels that the image actually snaps into much clearer focus. So this kind of got us thinking about genome sequencing as a tool to get 100% of the pixels in focus. Mirror VNTR is just looking at a fraction of the genome. Whole genome sequencing looks at the whole genome. So that is what gave rise to, back when we were doing that original 40 TB genomes project a decade ago, this emerging field of genomic epidemiology, which is simply reading the entire genome of all of the isolates from a particular outbreak to try to look for clues about who transmitted to whom. And the simplest way of explaining it is with the analogy of the children's game of telephone, where you've got kids in a line, somebody whispers a sentence to the first kid in line, who then whispers it to the next kid, to the next kid, to the next kid, and by the time the sentence reaches the end of the line, it has changed. It's mutated as it spread from kid to kid to kid. The same sort of thing is happening to the Mycobacterium tuberculosis genome as it's transmitting from person to person in an outbreak. So what we're doing with genomic epidemiology is really the equivalent of doing this kids game, whispering a sentence and then telling the kids, just go shuffle yourselves around the room randomly. Go to any old spot in the room. Then you go around to each kid in turn and you say, okay, what did you hear? What sentence did you hear? What did you hear? And from what they tell you, you try to figure out the order that they were sitting in. So if the word cat changed to the word bat in some of the sentences, you'll be able to determine that the kids that all heard cat were sitting in one part of the line together, the kids that heard bat were sitting in another part of the line together, and right away you've managed to divide your outbreak of children into two separate groups, the group that heard cat and the group that heard bat. So really, what we're doing with genomic epidemiology is just that, but writ on a much larger scale. We are, instead of asking the kids, what sentence did you hear, we're sequencing the MTB genome from a particular patient. And the mutations in that genome are just like the mutations in our sentence. And we look for shared patterns of mutations across particular individuals to figure out within one large outbreak, what are the smaller subclusters that might help us to refine transmission further. So if we go back to that mirror-VNTR fingerprinting diagram and we sequence all of the isolates that share a mirror-VNTR genotype with at least one other isolate, we get a phylogenetic tree that looks like this. Here the red bars indicate samples that were taken from people born in Canada. Gray bars are samples, MTB genomes from people born outside Canada. And the black dots indicate those isolates in our data set that happen to be multi-drug resistant. If you use this data to give you a much more accurate estimation of the true rate of transmission or clustering of TB and BC, your estimate of clustered cases drops from 42% with mirror-VNTR genotyping to only 24% with whole genome sequencing. In other words, 1 in every 4 cases of TB and BC is transmitted to person and is potentially preventable. So just to give you an idea of what that data looks like, this is the data from one particular outbreak. Each row in this multiple sequence alignment corresponds to a Mycobacterium tuberculosis genome from a different patient. And the MTB genome in total is about 4.4 million letters long. But over the course of a multi-year outbreak, which is what you're seeing here, there's only a handful of positions, maybe 15 or 20 where mutations actually happen. So we can ignore the rest of the genome that's unchanged across all of our outbreak isolates, and we can focus our attention just on those interesting positions where there's variation, and that's what you see here. And visually, right away with this little coloring scheme, you can intuit that there are particular groups of patients who have identical or near identical isolates to each other, but are just different enough from some of the other patients that you can say, okay, well, maybe these people didn't directly transmit to each other. So to walk you through this particular example, these isolates at the top of this multiple sequence alignment were ones that were diagnosed around 2001 to 2004. We have done some historical sequencing back further than 2005, and we see lots of diversity here. TB mutates at a rate of approximately half a mutation per year. So if we're seeing mutations at, say, 10 different positions, that suggests that that strain's been circulating for at least 20 years. It's been going around for a while. If we look at this little group down here, all of these were cases that were diagnosed in one particular part of British Columbia. And you can see they're almost virtually identical. There's just this one interesting position where an A in some isolates turns into a C in other isolates. So we can take that group of individuals and suddenly divide them into two and saying, well, if you have the C mutation, odds are you received your infection from somebody else with the C mutation and gave it to somebody that had a C mutation. These were some interesting isolates too. They were separated by thousands of kilometers and 4 years apart, but they're only 2 mutations different. But it turned out when we dug into that story a little bit further, they actually happened to be in the same place at the same time many years before and would have received their infection at the same time from the same source. So this is the type of data, what it actually looks like. The type of analysis that we do on this, I won't go into great detail on because it's pretty mathematical, and I know it's pretty early in the morning when you're watching this presentation. But we've done a lot of work over the years on combining evolutionary and statistical models for identifying who transmitted to whom just from that genomic information alone. So there's a lot of math that goes into it, but we can tell not only just who transmitted to who, but likely when that transmission occurred as well. And we did this with a second large outbreak that we investigated. Remember that TB has that latent phase, so you might be infected and take many years to progress to active disease, or you might develop active disease right away. So with genome sequencing, we're able to answer questions like, okay, for every case in the outbreak, and this was an outbreak that got up to nearly 50 cases, when did that individual receive their infection? When was the actual outbreak happening? And this is really useful and interesting information for public health. It helps tell us when transmissions stopped, and that in turn allows us to declare outbreaks of TB over. So this is some work that we've been continuing on with for the last few years. This is the most recent paper that came out from that last year in Molecular Biology and Evolution. Again, just figuring out automated ways of reconstructing transmission events just from genomic alone. So a really powerful tool for understanding TB epidemiology, even if you can't necessarily get out there in the community and ask. So I'll finish with one very quick story, a paper that we published earlier this year in JID looking at pediatric TB cases, TB in children in BC. So we had, over that retrospective study period, there were nearly 100 cases of pediatric TB, and we had sequenceable material for half of those. We also had lots of really interesting information like where was that child born? Where were their parents born? If they were born outside of Canada or their parents were born outside of Canada, when did they immigrate to the country? Did they have a travel history of going back to, say, their parents' birth country for extended vacations? And we had this large data set of all these genotypes and all these genome sequencing results. So what we found when we used genomics to answer the question, where did children in British Columbia get their TB from, is we found it was very dependent on how old that child was and where they and their parents were born. So in the subsequent slides, the color scheme here is going to be kind of burgundy to represent children that were born in Canada to parents who were also born in Canada. The pink bars, lighter pink, are children that were born in Canada whose parents were born outside Canada, and blue are children that were not born in Canada. They were born overseas. So in the age distribution of cases, you can see that Canadian-born cases tend to be younger. Foreign-born cases of active TB tend to be teenagers. And where contact investigation and field epidemiology had suggested a potential source of a child's TB, whole genome sequencing confirmed that. So in our Canadian-born children with Canadian-born parents, it was often visitors to the home, community members, or sometimes adult household contacts. For almost all of our children that were Canadian-born with foreign-born parents, it was an adult household contact, often a parent that genomics indicated was the source. And we did have one child that was born overseas who was transmitted to in BC from an adult household contact. So in total, of the 49 pediatric cases that we investigated, whole genome sequencing suggested that 15 of those were the result of transmission in BC. And the rest of those kind of varied. We had Canadian-born children whose parents were born overseas, and those kids traveled back to those countries, so they probably acquired their TB abroad. We believe that almost all of the foreign-born children acquired their TB before they immigrated to Canada. And there are a couple of mysteries, too. So genomics can't solve everything. We can only sequence the isolates that we have access to, which in our case were the ones from BC. But it can tell us some really interesting things. And so to us, that suggested that one of the things that we do when we diagnose a pediatric case of TB, something called reverse contact investigation, we don't necessarily need to do that if we see an older child who was born overseas, because the genome sequencing work tells us that they probably didn't get their infection in BC. It was probably the result of acquisition prior to immigration or as a result of travel. So this is a really small but interesting example of how genome sequencing can really kind of change how we do our TB care. So what are we doing next? We're still looking at that retrospective data set as well as analyzing all of our new data. We've got a very cool project where we're looking at our transmission networks. We've divided people into people that transmit and people that don't transmit, and we're combining that with some linked administrative data we have to answer some questions, like are there particular clinical or epidemiological characteristics that mean that somebody's more likely to transmit TB than somebody else? And are there features of a small TB outbreak that would allow us to predict whether that outbreak is going to grow and become a particular problem later on? So this is some work that we're doing with machine learning and predictive analytics. So to sum up, TB is a really interesting organism to work in. It's got a really straightforward genome to analyze with whole genome sequencing, but we can ask and answer a lot of really important questions around diagnosis, around treatment, around transmission. And when we use that information that we're deriving from whole genome sequence as the basis for changing how we do TB practice, TB policy in British Columbia, we really think we can progress towards elimination. So lots of people contributed to this work, including folks at the BC Centre for Disease Control and the BC Public Health Lab, collaborators up in Yukon Territory, collaborators at Oxford University and Public Health England and around the world. It's been a tremendously exciting project to work on, and we're really excited to see where our TB genome sequencing analysis takes us next. Thank you all so much for joining in, and I'm happy to take your questions. Please do send them on to me. Thanks again.
Thank you, Dr. Gardy, for that informative presentation. And we will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appear on the screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question, Dr. Gardy, is what role can genomics play in higher TB incidence settings?
Yeah, that's an excellent question. So the work that I presented here is largely in these low incidence settings, places like BC, and we have very different needs and very different end goals than a higher incidence setting. So while I think the priority for high incidence countries really does need to be on simple, rapid case identification and getting people on antibiotic therapy right away, just using simple laboratory techniques, you don't need whole genome sequencing for that. What I think it's going to do is a couple things. I think, first of all, as we start to sequence more and more TB in low incidence settings, that's going to tell us a lot of stuff about the TB genome that we can use to develop better, rapid, non-genomic point of care testing for higher burden settings. So we're just going to be generating a lot of knowledge that's going to help us do better diagnostics for lower and middle income countries. I think there's also some interesting things that genomics can do. We know that in certain high-burden countries, reporting of TB cases isn't always perfect. The actual number of TB cases usually far exceeds the number that are reported to the government. And with some of the population genomic analyses, where we're using that evolutionary biology and the mass modeling, we can actually look at TB genomes sampled from a population and estimate the true population size. You know, we only sequenced 1,000 TB genomes, but based on the diversity we see, how large is the true population that's out there? So I think that can be quite informative in terms of saying, okay, is this country really on target with their reporting, or is there a lot of undiscovered TB out there that maybe we need to come up with better programmatic ways of identifying? So while I don't think whole genome sequencing is going to become routinely used in high burden settings anytime soon, I think there's a lot that we can learn from its deployment elsewhere that can feed back and result in better policy and practice in those settings.
Thank you, Dr. Gardy. And this next question is actually two parts. What about mixed infections or within host genetic variations? How does that affect your results?
Yeah, that's a great question. So because TB has this latency period, because it's a chronic infection and hangs out in people's lungs for a while, you might expect that the TB bacterium within an individual, the population of TB bacteria, might have a little bit of variation in there. So we've been working on some methods to account for the fact that, what if one person harbors, two types of TB, one where it's got a mutation at a particular position and maybe another where that mutation doesn't exist. How you then infer who they might have transmitted to, because some people are going to receive one variant, some people are going to receive the other variant. So we've been working to solve that with a bunch of different mathematical models. The other question, the other part to that, the mixed infection, we know from genotyping studies that in high incidence settings, as many as 1 in 5 people can be infected with 2 different strains of TB at the same time. This doesn't really happen in lower incidence settings like British Columbia, but it is a problem in higher burden settings. And fortunately, when we do sort of deep whole genome sequencing, we can actually bioinformatically separate out those 2 strains. We can identify mixed infections and we can analyze each one of those populations separately. It's a very, very new area of research. There's just been a few papers on it this year, but yeah, the sequencing technology allows us to kind of deconvolute those.
Thank you, Dr. Gardy. And we have one more question. Will genomics completely replace the current protocols in the TB laboratory?
Yeah, so I think in places like Public Health England, you've had almost a near complete switchover. The only thing that's really still happening is phenotypic drug sensitivity testing. And with some results that us as part of this large international consortium have coming out soon, I think in certain settings where the prevalence of drug-resistant TB is extremely low, I think within 2 to 5 years, you might see whole genome sequencing completely replacing all of the traditional laboratory steps. You would only do things like phenotypic drug sensitivity testing if the predicted resistance of your isolate suggests a certain treatment regimen, but then your patient isn't responding. There's some sort of empirical evidence that the treatment isn't working. Then you might go in and do phenotypic DST. But I think in certain settings, we are looking at a future where whole genome sequencing could be it. As I said earlier, certainly not in higher burden settings, lower middle income countries, but in certain state and regional and national reference laboratories, I think it's a very, very real possibility.
Thank you. And Dr. Gardy, would you like to provide our viewers with some closing remarks before we go?
Yeah, absolutely. This is one little slice of a fascinating world of public health genomics. I think TB is one of the areas where, because the TB genome is relatively straightforward and because it's such a critically important pathogen in terms of global public health, it's certainly been one of the ones that has been most studied with the genomic approach. And we've laid a lot of really interesting sort of theoretical and practical groundwork here. But the stuff that we've done in TB is totally extensible to other pathogens. And as we're starting to, as a public health and microbiology community, really trying to get a handle on what it seems like sort of increasing numbers of epidemics. We've got hospital-acquired outbreaks and superbugs left, right, and center, foodborne illness outbreaks happening regularly, emerging infectious diseases in the viral space, more and more Ebola outbreaks, Zika virus. All of these sorts of techniques are applicable across that array of pathogens. So it's a really exciting time to be somebody who's able to combine bacteriology, virology with genomics, with computational biology, and with epidemiology. And I think it's going to be one of the most interesting and most productive areas of biomedical research in the coming years. So it's a great place to work. You'll never be without a job.
Thank you so much, Dr. Gardy, for your presentation and your important research. We'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through December of 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's event. That's all for now, and thanks for joining us. Hope to see you again soon. Bye-bye.